Okay, so good morning or good afternoon, everyone, depending on where you are. My name's Trevor Dummer. I'm the National Scientific Co-Director of CAMPATH, the Canadian Partnership for Tomorrow's Health. And welcome and thank you for joining today's CAMPATH Trainee Research webinar. This is actually our first webinar of the new year, so especially pleased to welcome you all back. I see a few are still joining, but I think we'll get started now as it's at the top of the hour. Especially pleased today to be focusing on student-led research. The central aim of CAMPATH is to provide high quality health data sets to support training of the next generation of researchers. So it's really exciting to have two trainees along to talk about their research. Before we do launch into the presentation, I do want to acknowledge that I'm joining you today from the unceded territories of the Coast Salish peoples. This includes the territories of the Musqueam, Squamish and Tsleil-Waututh nations. I would also like to acknowledge that the traditional territories of the Mississauga of the New Credit First Nation and the Lenape, Wendat, Huron, Haudenosaunee Indigenous peoples on which the CAMPATH National Coordinating Centre based at the University of Toronto now stands. This territory is subject to the Dish with One Spoon, One Put Belt Covenant, which was an agreement between the Iroquois Confederacy of the Ojibwe and the Allied Nations to peacefully share and care for the resources around the Great Lakes. We acknowledge that CAMPATH teams work across Canada from coast to coast. Many of you are joining from coast to coast as well. We acknowledge the ancestral territories that are home to many Indigenous peoples from across Turtle Island, and we are grateful to have the opportunity to work on these lands. So as I said, this is a webinar on student-led research, and today we're going to hear from two trainees, both based at the University of British Columbia School of Population and Public Health, looking at things related to the built environment on cancer risk and also how improving methods to predict breast cancer risk using metabolomic data. So we have two speakers. Our first speaker, Atul Aravindakshan, is a PhD candidate at University of British Columbia School of Population and Public Health. Atul is under the supervision of myself and Dr. Parveen Bhatti. His doctoral research is studying the effects of residential greenness, walkability, nighttime light and air pollution on the risks of breast, prostate, lung and colorectal cancers. So as I mentioned, Atul is a PhD candidate. Trinh is a master's student also at UBC in the School of Population and Public Health, and she is under the supervision of Dr. Parveen Bhatti. Lee's thesis is looking to predict future breast cancer risk and identify associated metabolomic biomarkers. So two really exciting projects related to cancer and cancer prevention. As many of you know, if you've joined these sessions before, we'll have the trainee presentations. We'll have time for some questions as well. Atul will go first, followed by some questions to Atul, then Lee will follow him. Please use the Q&A button at the bottom to put your questions in, and we'll deal with them when we can. Okay, so without any further ado, I'll hand you over to Atul.
Thank you, Dr. Dummer, and also thank you, Megan, for organizing this this webinar. I appreciate the opportunity to talk about the research. So my research is using CAMPATH data. And
sorry, Atul, I think you need to share your screen.
Yes, I do. I haven't started yet.
Oh, sorry. My apologies. I didn't want to interrupt.
No, that's absolutely fine. So my research is using CAMPATH data, but I'm still in the early stages of the analysis, so I don't have any results to share today. But I'll be talking about the background, the objectives, a very brief introduction to the statistical methods, so on and so forth. Before I begin, I just wanted to acknowledge that the place where I live and study on UBC campus is located on the traditional, ancestral, and unceded territory of the Musqueam people. It's been a place of learning, and I'm grateful for the opportunity. So with that, I'm just going to share my slides. I'm assuming it can be seen. It's all good.
Perfect.
So as mentioned, my research investigates the causal effect of the built environment on cancer risk and its association with metabolites. A bit of background about cancer and metabolites. So cancer is the #1 cause of death in Canada. If you look at lung cancer, the 5-year survival rate is about 19%. there is an improvement in the survival for breast cancer over the past decades, but we find that this improvement is essentially seen in people who are diagnosed at an early stage. Talking about the incidence, breast, prostate, lung, and colorectal cancers are the top 4 most common cancers in Canada. Regarding colorectal cancer, there's in fact an increase in the incidence in younger populations. Added to this fact, Canada has an aging population demographics and is expected that we will see an increased burden of cancer on healthcare. So prevention is essentially better than cure. And this morbidity and mortality of cancer can be mitigated by interventions which reduce or alter exposures to risk factors. Quite often we find that these interventions are geared towards individual level behavior changes. Don't smoke, reduce alcohol intake, increase physical activity, increase your fruit and vegetable intake. But they can be ineffective if we don't account for socioeconomic and cultural aspects of the population. If we move to an ecological level, the built environment can be modified to influence individual behaviors, and thereby it can alter cancer risk. When I talk about built environment, I'm talking about residential buildings, the road network, the pedestrian network, air pollution due to human activity, the light at night, so on and so forth. So it's known that this built environment can alter health or affect health through dietary choices, physical activity. If an area is more walkable, you're bound to move around more. The light at night can affect circadian rhythms, hormonal changes, so on and so forth. But despite all this knowledge, the actual effect of the built environment on cancer risk is still unknown. We do know that air pollution is a group 1 lung carcinogen, but given that Canada has a low level of air pollution, it's still worth exploring whether this association still holds. So these unknown pathways and these unknown mechanisms can be studied through traditional epidemiological methods where we have an exposure, we have an outcome, and we look at the risk estimate in a particular population. But we can also look at biological markers or biomarkers. As the name suggests, biomarkers are molecules in living organisms, and they are used to study disease processes. I'm looking at metabolites, which is an example of a biomarker. And they are essentially the end product of DNA processes. So any change in the DNA results in a change in the metabolites. And through that, we can have an indirect estimate of any progression or any an indirect estimate of cancer risk. So based on this information, I have 3 objectives. The first one is to study the effect of greenness, walkability, nighttime light, and particulate matter and nitrogen dioxide on the risk of breast, prostate, lung, and colorectal cancers. The second objective is to measure the direct and indirect effect of greenness on cancer outcome. So on the left-hand side, you can see an arrow going from greenness directly to cancer outcomes, which is essentially the direct effect. There can be some mechanisms in between, but I'm not aware of those. So as of now, at this point of time, I'm assuming that to be the direct effect, and exposure to greenness should essentially reduce cancer risk, ideally. But there's also indirect effects where greenness can affect the walkability in an area. If it's natural vegetation, the walkability in an area decreases. If it's a park, there's a built walkway which helps in moving around. This in turn can affect physical activity, diet, other things, which ultimately affect cancer outcomes. Similarly, greenness can also affect air pollution and nighttime light due to human activity, which in turn affects cancer outcome. So for the second objective, my goal is to essentially estimate how much of the effect is the direct effect and how much of the effect is due to the indirect effect through walkability, air pollution, and nighttime light. For the third objective, I'm investigating the association between greenness and, I'm investigating the association between all the exposures mentioned previously and metabolites. I mentioned that the metabolites can provide an indirect estimate or an estimate by extension. So it essentially just replaces cancer in the diagram. Coming to the data sources, CanPath has rich data. It has data from 7 provinces and, 7 cohorts and 10 provinces. I'm using data from the British Columbia Generations Project and the Alberta's Tomorrow Project. I have around 69,000 participants from both cohorts. The cancer outcomes are linked through the cancer registries for each province. There's also environmental data from the Canadian Urban Environmental and Health Research Consortium, also known as CANU, and the data is linked to the participants. For CANU has a lot of environmental exposures, but I am particularly looking at the ones previously mentioned, that is greenness, air quality, nighttime light, and walkability. I also have walkability linked from Where Matters study. It has a walkability index for 2006, 2011, and 2016. And the data is linked to residents in Metro Vancouver, located in British Columbia. So this is where researchers typically have to pay a little bit of attention when combining cohorts, try and figure out which data is linked at what time point to which cohorts. To give another example, if I'm talking about the British Columbia Generations Project and the exposure of greenness, the data is available from 1984. So if the person was alive and the data is available, I would have an observation for 1984, 1985, 1986, so on and so forth. But if we talk about ATP, the data is available at baseline and any follow-up. So it's more active based on the address reported at that point in time. The information is available only for that address reported. Finally, I have 233 metabolites, which are linked to participants in British Columbia Generations Project. It's about 1,320 participants, 1,320 participants. The participants were non-smokers and were not pregnant at the time of assessment. So there was a lot of information given earlier, but just to clear the slate and kind of focus on the main point, I have 6 exposures. One is greenness, 2 measures of walkability, one from CANU, one from Where Matters, light at night, and air pollution metrics of particulate matter and nitrogen dioxide. For the first objective, the outcomes are breast, prostate, lung, and colorectal cancers. For the second objective, I'm looking at the direct and indirect effect of greenness on cancer risk. And for the third objective, I'm looking at the change in metabolite concentrations depending on exposure to the environmental factors. For the statistical analysis, cancer risk, or for the first objective, I'm using a Cox regression model or a proportional hazards model, and I'm looking at the association between each exposure and each cancer using age as the time scale. The covariates and confounders will be selected through previous research and change in effect estimates. As a sensitivity analysis, I will be looking at the exposures on the continuous scale as well as on a categorical scale. Coming to the mediation analysis is contingent on significant findings from the first objective. If there is no effect of greenness on any of the cancers, then there's no point studying any direct or indirect effects. But assuming that there are some significant findings, I will be doing the analysis based on the counterfactual method. Essentially, this involves building a model with cancer as the outcome, greenness as the primary exposure, and the other environmental exposures as mediators. This will be followed by building other models with the environmental variables as the outcome and the greenness measure as the exposure. Now, based on the results from these multiple models, there will be a combination of the coefficients, and we'll be able to calculate the direct and indirect effect of greenness on cancer risk. The last and final objective is a cross-sectional analysis. It's restricted to the 1,320 participants in British Columbia. I'll be looking at the association of each and every environmental risk using linear models. and estimate the change in metabolite levels. The metabolites will be log transformed due to assumptions of normality. Strengths and limitations. So it's a cohort study. It's longitudinal in design. So it improves causal associations or improves the belief in causal associations. The exposure is definitely measured prior to the outcome. We have a lot of exposures from both canoe as well as wear matters, and approximately a size of 69,000 individuals from British Columbia and Alberta. This is expected to drop a little just because there would be participants with prior cancer and some missing data, but I'm not expecting a huge drop. Finally, coming to the metabolite analysis, it is exploratory in nature, and it's able to provide insight for larger scale studies, which will be conducted in the future. The biggest limitation, I would say, is the non-probability sampling. So participants essentially volunteered for the study. So that means they had, it means if you look at the distribution of British Columbia and Alberta, or the cohorts in British Columbia and Alberta, you'll find that majority of female, majority were from high-level education status and high-level income status. Over 90% of the participants in both cohorts were Caucasian. So the distribution's a little skewed, but just because of the in-depth questionnaires, which has assessed many habits and many exposures, I do believe that we'll be able to look at the biological effect, which is seen, and we'll be able to capture that in our study. Coming to the metabolite analysis, our study does not investigate temporal changes. I mentioned that it's cross-sectional in nature. And also, majority of the metabolites assessed are focused on lipids. I did mention that the participants were non-smokers and were not pregnant at the time of assessment, so that limits generalizability. So the study has the potential to inform land use, urban planning, and it's of interest to stakeholders and policymakers who are involved in urban city planning. The Where Matters project, in fact, is already publishing it's already publishing a policy briefing for a few stakeholders such as Metro Vancouver and North Vancouver. We also expect to publish all these findings and all these results in research studies and research articles. And we expect that these findings would inform any studies conducted in the near future. And with that, I would just like to say thank you to my supervisors, Dr. Trevor Dummer, Dr. Parveen Bhatti, and also my committee members, Dr. Rachel Murphy and Dr. Lawrence Frank. The study is made possible through funding from the CIHR Canadian Graduate Scholarship. And last but not the least, participants and staff at BCGP, Alberta, Tomorrow's Project, and the Canadian Partnership for Tomorrow's Health. Thank you. Thank you for listening.
That's great. Thanks so much, Atul. That was a fantastic, nice and clear presentation. Really exciting to see you getting going, and hopefully we'll see you back in a year's time to present some findings. So, got some questions in the Q&A. So please do remember, if you any questions for Atul, we've got about 10 minutes for a Q&A for this component. So any questions, please use the Q&A button in the bottom right-hand side. So the first question, this is great work. Wondering how you can plan to take into account genetic risk factors for these cancers, such as the BRCA for breast cancer, prostate cancer.
Yeah, that is actually a good question. It's kind of a limitation of the study because I don't have any access to genetic data. There's a lot of ethical considerations when we have access to that, and we don't have access to that at this point of time. So I won't be able to assess for that, but essentially, yes, it's a limitation of the study.
Okay, thanks. Very interesting research. Could you please explain how you will measure and quantify greenness? What is NDVI?
So there are different measures of greenness. One of them is the normalized difference vegetation index, also known as NDVI. It's essentially obtained through satellite imagery, where we look at the different spectrums of light. So there's the red and infrared spectrum of light. And the way it's calculated, it's the difference between the near infrared band and the infrared band divided by the sum of the near infrared band and the infrared band. Uh, I think I can share my screen for the formula. So this is essentially how it's calculated. And what happens is all based on satellite imagery. I am using just the ratio which is given to me through Canoe, and it's linked from 1984 till I think 2016 it's available. It doesn't take into account the different types of greenness. So for example, if there's park count or even the different types of trees, if it's a shrub or if it's a big tree, for example, it's not able to account for that, but it's just a surrounding greenness in the residential, in the vicinity of the residential address given by the participant.
Great, thanks. I just had one follow up on that. Can you expand a little bit? You mentioned it's not parks, it's not trees. What's the limitations of NDVI compared to other potential measures that you didn't have access to?
To give an example, if the type of greenness essentially, okay, I'll just start a little bit. So essentially my theory is that the type of greenness affects a person's physical activity. It affects a person's access to dietary choices, so on and so forth. So if it's a natural vegetation, that becomes a little difficult for a person to move about. But if it's a park, it becomes easier. There's typically a walkway. Person can walk from point A to point B more easily compared to natural vegetation. So the different types of greenness can affect the behavioral patterns in a person. And that is ultimately supposed to affect the cancer risk. This measure of NDVI does not take into account if it's a park or if it's a forest or if it's just a lawn, for example. So one of the limitations is it's just it's one of the limitations for assessing NDVI.
Yeah, that's great. Thank you. Got some more questions here. So this is a two-part question. It seems that you expect that metabolites will become a surrogate for lung cancer or a marker of cancer risk. Is there evidence that known patterns exist?
There is evidence. It's still mixed. There aren't any metabolites which are, or rather, I've not come across any metabolites which we can say for certain that this is what affects lung cancer or this is what affects prostate cancer. But what we are doing is we are looking at the effect of the association of the built environment on metabolites. And once we get those associations, I'll be looking at the metabolites which pop up or which are significant in the study. And I'll be looking at known biochemical pathways which are connected to the different cancers and try and see if there's any connection or if there's any pathway which can be singled out and find. This is exploratory in nature. I mentioned that there are only 13, 20 participants. So it's not a full-fledged study. But the idea is that once we get those pathways, they can inform future studies and it can be built on that.
And another quite technical question from the same. When modeling risk, how will you handle overfitting?
So I'm not looking at predictive modeling as such as of now. I'm looking at causal modeling. So my focus is on accounting for any types of confounders and of course known predictors. But I'm not focused on handling overfitting. It's more about the causal association between the built environment and cancer risk.
Great. I think we should have time for the final. There's two more questions here, so we'll get through these two, I think. Awesome job, Atul. How did you choose the variables you've included in your DAG? Are there any variables in the data that you did not include?
So in my DAG, I typically looked at the variables which are considered in previous research. And of course, there were variables which are interested, for example, greenness, walkability, so on and so forth. And I looked at associations which with the primary exposures, and if any of those habits or any of those associations can be linked to cancer. So it's all based on previous research, essentially. Are there any variables in the data that you did not include? As of now, I did mention some variables about medication and current health status. They might not be included based on limitations in the data. It depends on how much missing data is there. So I'm still cleaning the data and going through it, but essentially everything listed on the tags, I do have access to that data. It's a matter of how much data is available to me and whether it fits the model appropriately.
Yeah. Okay, and one last question, I think, for you. Very interesting work. Would you be able to expand on how you're measuring air pollution?
Air pollution data is again provided by Canoe. They have different measures. So there's particulate matter, there's nitrogen dioxide, there's sulfur dioxide, and ground level ozone. These are mostly considered as traffic-associated air pollution metrics. Some of them use land use regression, which is basically predictive modeling for calculating the exposure at a particular zip code or postal code. What happens is that there are sensors placed around the country, and these sensors are sample the air quality around that area. And then with a combination of those measurements and satellite imagery and predictive models are used to estimate the exposure based on zip codes or dissemination areas.
That's great, thanks. So I think I see no more questions, and we're almost at the half-hour mark, so we'll be moving over to Lee's presentation now. But if anyone has any further questions they want for Atul, please type them into the Q&A. We can either handle them at the end or we can do them offline. So if questions do come to you further on, please do use the Q&A. So now I'd like to hand you over to Lee Trinh, who's going to talk about her master's thesis research. So Lee, over to you. Thank you.
Yeah, I'm going to share my screen real quick. Here you go. I'm
Lee,
using my full screen.
Yeah, you might need to just switch it. That's it.
Oh.
I think.
Sorry. Yeah. Let me actually. Does that work?
That's perfect.
Okay. All right. Sorry about that. All right. Thanks, everyone. And thank you to CanPath for giving me the opportunity to present my master's thesis, which is on pre-diagnostic serum metabolomics and breast cancer risk. So I'll just be giving an overview of breast cancer epidemiology and metabolomics. So breast cancer is the most commonly diagnosed cancer among women in Canada, and it accounts for about a quarter of all estimated new cancer cases. And breast cancer also accounts for the second most common cancer deaths in Canada. It accounts for about 13% of all cancer deaths, and it's only second to lung and bronchus. Now, incidence and mortality of breast cancer have largely stabilized over the year, but survival is still pretty poor, especially for the later stages. Stage 4 survival, for example, is only at 20% at the 5-year mark. In addition to that, a lot of breast cancer survivors also face many long-term health effects, such as osteoporosis, fatigue and neurological dysfunction, respiratory infections. And they're also at higher risk of cancer recurrence and second primary cancer. So all of these lower their overall mortality and quality of health. And also just the sheer number of breast cancer patients putting a huge strain on the healthcare system. So in light of all of these, cancer prevention remains a critical goal. Now, research has shown that unfortunately, large population-wide intervention strategies are not effective in the long run. And effective strategies require usually more intensive resources because it requires constant supervision and social support and regular reporting. So an alternative, or I guess an adjunct to this idea, is that if we can somehow identify or can assess breast cancer risk from the general population before cancer development, and we identify people who are at elevated risk for breast cancer, we can then target these people for more intensive interventions and enhanced screening for earlier detection. And that will help improve treatment outcomes, hopefully. So that's where the idea of metabolomics comes into question. So Atul has defined kind of this field of studies and how it can be potentially a very promising reflection of the biological processes that facilitate cancer development, because it's both the downstream product of the DNA transcription process and also a product of a lot of environmental biological processes that are changing over time and affected by the environment, like aging, like hormone-related events, and also diet and lifestyle factors. So the idea is that if we could take blood samples from the general population, especially pre-diagnostic samples, and profile them to understand the contents in their samples, we could potentially understand what are associated with elevated cancer risk and also use those samples to gauge people's future breast cancer risk. So that leads us to my study, which aims to determine metabolomic signatures that are associated with breast cancer risk, and then utilize these metabolites to predict future risk. Now, so this is a schematic of my study design. I'll go into more details in later slides, but the idea is that we're using data from two CanPath regional cohorts, the BC Generations Project and the Alberta's Tomorrow Project. And from each of these cohorts, we ascertain cases that are cancer-free at baseline, but then go on subsequently to develop breast cancer. And then these diagnoses are ascertained through cancer registries, and these are through annual linkages with CanPath data. And then for each of these cases, a control is matched on a 1:1 ratio from each respective cohort. Each case is also matched to a control based on age of blood collection, year of blood collection, and baseline menopause status. And then, so for all these participants, we use two different types of data. The first one is the health and lifestyle questionnaire that each participant has to fill out at baseline enrollment. And this questionnaire includes measures like demographics, like age, ethnicity, education level, income level. We also get an insight into family and reproductive history, such as the number of first-degree relatives with a history of breast cancer, age at menopause and menstruation, pregnancy, childbirth, breastfeeding, also usage of hormonal contraceptives and hormone replacement therapies. And we also have lifestyle behaviors like BMI, alcohol consumption, and smoking status. And then the second type of data that we used is metabolomics, more specifically serum, non-fasting serum metabolomics. So these are the samples that are also collected at baseline enrollment, and then for our study and stored in -80 degrees. So, and then for our study, they're shipped to General Metabolics, which is a company in Boston, Massachusetts. And they do the metabolomic profiling for us using a technique called quadruple time of flight mass spectrometry. And I'm not going to go into details about how this technique works, but if you want to know, let me know and I can try my best to explain through it later. And then but what that process does is that it, using for each sample, it identifies a spectrum of different metabolite levels for something called a mass per charge ratio. And then the idea is that those correspond to unique compounds, and we can reference them against known databases to identify the names of these compounds. And in our case, we reference them against 3 different databases, which are HMDB, CHEBI, and KEGG. And then with all of this data acquired, we get to the statistical analysis. And so we have 2 primary goals, which are, again, assessing breast cancer risk and breast cancer prediction. So in order to assess breast cancer risk associated with each metabolite, we used unconditional logistic regression, adjusting for matching factors and any confounding that are present. And then for breast cancer prediction, we trialed a few different machine learning classification methods, then including lasso, random forest, and partial least squares discriminant analysis. And then as a secondary analysis, we repeated these two steps for different subgroups, which are postmenopausal participants, participants with cases of ductal carcinomas, and participants with hormone receptor positive cases. So those are the methods. As for the results, so this is a summary of the study study participants in our project. So I think this is something that Atul found in his research as well, but the most health and lifestyle characteristics are very similar between study groups, and there's not a lot of differences between cases and controls. The only notable difference is that cases are more likely to have first-degree relatives with breast cancer, and we know this is a huge risk factors that are determining future breast cancer risk. Other than that, overall, they're very similar. And then results for the metabolomics profiling, 854 metabolites were detected in at least 50% of our experimental study samples. And because of the peculiarity of the quadruple time of flight mass spectrometry method, the most of these compounds have multiple identities that they could be associated with, because some of them might be differently charged ions, and I can also go into that later if you're curious about that. But essentially, so for some of these compounds that we identified, we're not fully able to know exactly what formula that associated with them. And then another thing that we ran into is that there is a different is that there is separation between BC data and Alberta data. So essentially, Alberta data showed a different grouping pattern from BCGP data, and we don't want that because then that kind of affects the quality of our data. So that required a bit of normalization as well. And now coming to the main goals of our study. So this is the odds ratio of 24 metabolites that were found to be significantly associated with breast cancer. And of these 24, 13 are associated with decreased breast cancer risk, and 11 are associated with increased breast cancer risk. As you can see here, a few of the metabolites do not have any annotations. So I just kind of left them as the identifier from the metabolites. So I have not been able to done the literature review thoroughly to understand if they have shown up in other breast cancer the breast cancer literature. But I've seen a few of, for example, these fatty acids that are theorized to be associated with increased cancer development through the cancer metabolism processes. So at least some of this evidence appears at this stage to be consistent with previous literature. But so far we have not been able to do more with that. And then, so for the next goal in the results, which are the risk prediction, we conducted a 10-fold cross-validation, and the results show that random forest outperforms LASSO and PLSDA, which is partially squared discriminant analysis, at all stages. So for this, we used, sorry, to back up a little bit, for this, we used metabolites that were significant after adjusting for multiple testing. And so we used metabolites, only 24 metabolites that were significant at the false discovery rate of 0.1. We used metabolites that were significant at a rate of 0.2, which increased it to about 78 metabolites, 78 inputs, and then all of the 854 metabolites. And then, so from this analysis, it looks like the cross-validated predictive performance is really high for random forest in all of these 3 different models. Whereas for LASSO and PLSDA, accuracy increases as the number of metabolite increases. And then afterwards, what we did is like, we had set aside a 10% holdout test set, which we did not use in any of the training or validation, but only for the last stage of the model performance testing. And unfortunately, in this stage, the best performing models from all 3 of these methods did not perform very well. And as you can see at the bottom there, that's the AUC statistic for the models. And essentially, they are only slightly better than 50%, which equals to random chance. So there's definitely more improvements and we might want to make these prediction methods more externally generalizable. And then just a quick note on the results for the subgroup analysis. A lot of this there's a lot of numbers, and essentially it just means that the metabolites here shown here are the ones that are significant in the full analysis and also show up as a significant metabolite in at least one of the subgroup analyses. And the strength of odds ratio is, the strength of associations are also pretty consistent throughout the different analyses. And so it shows that these metabolites do show do have potential promises in as biomarkers for these different conditions. And then in addition to that, we also actually observed associations for new metabolites that are only seen in the subgroup analysis, but not in the full sample. However, we are not certain of their true associations, or if they are just showing up as false positive due to the smaller sample size. So more investigation on that later. So just for the discussion for the next step in my project and the future directions that I think studies like this can continue on to be. So for the next step of my project, I'm going to conduct literature review to assess the biological significance of metabolites that are identified in the regression analysis. And then in addition to that, we may also try to fit predictive modeling for the different subgroups to see if to observe the predictive performance. And then in general, for future directions, we definitely need larger scale studies with more diverse participant samples. And I think a large prospective cohort study like HandPath would give a great opportunity for all this data. Sorry about this, but this, I wanted to say that we can just, we want to incorporate other methods for this kind of analysis, and that means different adjustments for risk factors, different types of omics studies, for example, a lot of multi-omics studies, and maybe explore different mass spectrometry methods as well. So there are also a lot of different directions that you can go from this like initial metabolomics study. So that is all I have for today. I would like to thank Dr. Parveen Bhatti and my committee members. I'd like to thank the staff at BCGP and ATP, the staff at General Metabolics, also CanPath participants for contributing their data to the study, and also funding from Cancer Research Society. And with that, I'll leave you with the questions.
Thanks, Lee. That was an excellent presentation. Very nicely presented, very clear, really interesting. So we've got some questions in the Q&A. You mentioned ductal carcinoma. Do you exclude women with invasive lobular carcinoma? If so, can you explain the basis for this?
Yes, for the subgroup analysis, where I decided to look into ductal carcinoma as one of the subgroups. For the full analysis, we do include both ductal, lobular, and mixed, and other types as well. It's just that we wanted to see if associations for these metabolites hold for the subgroups that are dominant subgroups in this population.
Great. You report differences in metabolite profiles between BCGP and ATP. Have you examined the pre-analytical factors associated with the samples? Does anything help explain the different profiles?
I really want to. We're still waiting for the data, the pre-analytical data from ATP, and especially because a lot of the variation is on specific samples from ATP. So it would be nice to be able to explore and account for all these unwanted variations. But so far, I don't have access to this data.
Great. In your machine learning with Lasso and RF, were all the metabolite measurements standardized?
Yes, they were standardized for both the logistic regression and for machine learning.
Great. Another hi Lee, great presentation. For the metabolite notes as 184 and 724, et cetera, do you plan to have metabolite class group name, if not a more detailed full name that matches to it? Were the majority of the metabolites associated with increased risk considered as fatty acids? So it's a two-part question. Were the majority of the metabolites associated with increased risk considered as fatty acids, or are you seeing trends that suggest certain exposures or biological mechanisms associated with increased risk?
Yes. So as part of the literature review, I definitely would like to label the classes of these different metabolites and then look into these different bigger groups, different bigger categories. I do see a few fatty acids that are associated with increased risks. However, we also see a few fatty acids that are associated with reduced risk. And so this is what happens a lot in metabolomics literature, which is we find a lot of this, we sometimes find a lot of the same compounds, but the direction of effect are pretty contradictory from study to study. So I'm expecting to see a few amino acids, a few fatty acids, a few lipids, a few hormones, but overall, haven't looked at that for my particular data, but yes, that would be something that I'm interested in doing.
Great, thank you. Another question. Thanks for your presentation. Just wondering, there were a lot of metabolites identified as associated factors in one or other direction. Would there have been any advantage to reviewing the literature ahead of running the assays, narrowing down the previously identified and associated metabolites to be tested?
That's a good question. So actually, there is a whole field of metabolomics that is dedicated to that, and that's targeted metabolomics. And the idea is that yes, you do a literature review and then narrow down your metabolites of interest to only those that have been explored in the past. So that is a very valid approach, and I think it is something that we can complement with our untargeted approach as well. For our study, this particular study is more a discovery-based study, so we would just like to look at everything and like use a larger scale of possible metabolites, just so that we don't rule out anything that it has not shown up before, but that has the potential to be a biomarker here.
Great. I had a question. I think that sort of partly answers that, talking about discovery. But so you've got this where you have sort of 24 metabolites that are associated, and 13 are in one direction, 11 in the other. If you take, how do we use that information? You know, what's the next step? You know, it's discovery, but if we're talking, because you talked at the beginning about prevention and risk factors, how do we use that information if some of it's contradictory and in light of what we know from other studies as well? So if you could talk a bit more about that, be very helpful.
Yes. So let's say if at some point during the future we come to enough robust evidence that some metabolites are associated with increased or decreased cancer risk in one direction or another, with enough like validated evidence, then hopefully the idea is that we would be able to sample patient blood through, routine checkups or any medical visits. And if we see increased signatures of these specific compounds in patient blood, then we can then signal them. We can then kind of gauge. We can then kind of gauge their risk in the population and say, oh, hey, this person may have increased breast cancer risk. We can look at their lifestyle factors, what are things that are attributed to these increased levels of metabolites, and maybe hopefully work with them to help lower these things through, modifiable factors. Yeah, things like that.
Great,
That's
thank
just
you.
an example.
Another quick question for you, Lee. So fascinating research. Just curious, is there a national or perhaps international database for these metabolites in public health research?
Yes. So the three databases that I mentioned are our HMDB, which is based out of Canada, CHEBI, which is based out of the UK, and then KEGG, which is based out of Japan. And they are metabolite databases that you can use to reference your measurements against. I am not I am not aware of anything that is specific to cancer research, if that's the question. But I would not be surprised. But I do know there is an exposome, there's a blood exposome database, for example, that is more relevant to metabolites that are present in biological, in blood samples, for example. So I wouldn't be surprised if other databases of that nature are already out there, are being built.
Great. We've just got a couple of minutes, so before we wrap up, and I want to make an announcement for the next webinar. But very quick question, with a short answer, but it's probably could be really long answer. You used 2 of the regional cohorts. What's the main challenge of bringing together those 2 regional cohorts? Given CAMPATH is a national cohort, but that's all mentioned as well. Some of the challenges of using 2 separate regions. So in 30 seconds.
It's just a lot of administrative stuff to go through, but the harmonized data is really nice.
Great. So it's the administrative side of things. That's fabulous. I think we will wrap up there. There's no more questions in the Q&A. So thanks, Lee. Thanks, Atul. Fantastic presentations. Thanks for everyone for joining in. I do want to mention before you all go, next Access Office open office hours is on the 31st of January, and this is actually a student-focused office hours as well. So this is particularly relevant for anyone here who's a trainee that's now thinking this could be a really interesting source of data for my study, or a supervisor perhaps who's thinking they've got a student that might want to be using some of these data. As many of you know, if you've joined these sessions before, CAMPATH has got data on over 330,000 Canadian participants, health-related data, lifestyle, linked environmental data from Canoe. So there's lots of data out there. Office hours is an opportunity to find out about the data and the administrative processes, which, as Lee's mentioned, there's a lot of things to go through. So the open office hours is about student-led projects and how to have a successful student-led data access request. So with that, I'll leave everyone to get on with the rest of their days. Thank you for joining our CAMPATH webinar. Thank you once again to our speakers. Really interesting to see researchers using the data and proceeding with some really interesting studies. So we'll see you next time. Thank you.
Thank you.
Thank you.